Hello everyone. Today I'm going to be teaching you about photosynthesis, which is the process that lets plants make food and provides oxygen for life on Earth. So, what is the purpose of photosynthesis? So, the purpose of photosynthesis is to use sunlight here, water, and specifically 6 water molecules, and 6 carbon dioxide molecules, which is also known as CO2, and we're going to turn that into food for plants along with oxygen. And this oxygen will be used by us humans and a lot more organisms. So let's look at the equation for photosynthesis. So as you can see here, this is the equation for photosynthesis. So the equation for photosynthesis, it wouldn't be an equal sign here. This would be an arrow to show that there's a transformation. And the equation for it is 6 CO2, which is carbon dioxide, plus 6 H2O, which is 6 water molecules. And again, this 6 CO2 is 6 carbon dioxide molecules, along with light, and the light will come from the sun. And this will become into C6H12O6, or which is also known as glucose, along with oxygen, which is 6O2. So it will create 6 oxygen molecules. So what organisms use photosynthesis? So a lot of organisms use photosynthesis. So, for example, it can be done by plants, algae, along with some bacteria known as cyanobacteria. So now let's look at the different types of photosynthesis. And there's going to be 2 main types. There will be light-dependent and light-independent reactions, and these 2 will combine to make sure that photosynthesis occurs. So let's now look at that. And make sure to remember that although there are light-dependent and light-independent reactions, these 2 combine to create photosynthesis, so these are not different types. So now let's start by looking at light-dependent. Alright, so this is a very detailed diagram here of light-dependent reactions. So first of all, light-dependent reactions happen inside the thylakoid part of the chloroplast. So as you can see, I showed the thylakoid right here. So it happens in these little bubble things inside of the chloroplast. This does not happen in the stroma. This is the stroma, the outside part, past the thylakoid, because that is where light-independent reactions will happen. So how does light-dependent reactions work? So sunlight is going to hit the chlorophyll, and it will split water, and this will release oxygen. And then there will be the excited electrons, as you can see right here. These are your excited electrons, and they'll help make 2 energy molecules, and these are ATP and NADPH. As you can see, here's your ATP, and this is your NADPH. And this happens because of the excited electrons. So now let's go a little more detailed with what I just talked about so you can get a better understanding of it. So as I already mentioned, sunlight is going to hit the chlorophyll pigment, and this is obviously found inside the thylakoid membrane. This light energy will excite the electrons that are inside, and they'll be passed around this chain of proteins, which are shown here. These are all proteins. And this chain is known as the electron transport chain. Okay, so this is your electron transport chain. These are, in basic terms, these are just proteins that transport your electrons so that it can create your NADPH and your ATP over time. So at the same time as this, water molecules are being split apart inside the thylakoid because, as I previously mentioned, one of the reactants for photosynthesis is water. So your water molecules are being split, as you can see. They're being split into oxygen. It's being split into other things along with electrons, and this is called photolysis. So it will make your electrons to replace these excited ones that you just did through the transport chain. You will have protons, which is your H plus right here, your 2H plus. This is your protons, and finally, it will release oxygen as a byproduct. So as the excited electrons move along the protein chain, they're basically pumping more protons, or your H plus here, into the thylakoid lumen. And what this is going to do, it's going to build up a high concentration of protons inside. And this will create an electrochemical gradient, which we'll talk about later. And then finally, these protons, they're going to go flow back out and whatever, and which we'll talk about more next. And this will create something called ATP synthase, which will kind of spin ADP and phosphate together, and this will create your ATP. So this is your phosphate, and this is your ADP, and together this will create your ATP. So in the end of the day, the light-dependent reactions will produce ATP and NADPH, and it will also release oxygen gas. And now, as you can see, it will just go out straight out the cell. It's just leaving, and it's also leaving the chloroplast. So now we're gonna talk about light-independent reactions. We're gonna talk about the electrochemical gradient. Okay, so now let's talk a little more about the electrochemical gradient. So this will happen inside the thylakoid lumen. So basically, when your sunlight is going to hit the chlorophyll in the thylakoid, it will excite your electrons, and these electrons travel across this electron transport chain, like I had previously mentioned. This is your electron transport chain. I did not mean to draw there. And this is a row of proteins in the thylakoid membrane. And as you can see, the arrows that it's traveling, the PS2 going to the PS1, and you can see these dots, these resemble your electrons, your excited electrons. So, these will travel along this chain, and as the electrons move, they power the proteins to pump protons. And again, if you remember, your protons are your H plus right here, and these are ions, and these will be across the membrane into the thylakoid lumen. Also, when your water is being split, more H plus ions, as you can see, it creates more H plus ions, and they're being added straight inside the thylakoid lumen, and this will create an electrochemical gradient. And this is a high concentration of H plus, or positive hydrogen, inside the lumen, and a lower concentration outside. And the purpose of this is to store energy, because the protons want to diffuse back out, but they can only do that by passing through a protein, which is called ATP synthase, which is right here. As you can see, I labeled this as ATP synthase. And as your positive hydrogen ions are flowing through the ATP synthase, it spins and it produces ATP. So as your ADP and your P kind of spin around, this will create ATP, and this is a chemical energy used by the cell. So now we're going to be talking about light-independent reactions, which are also crucial for the plant to get energy. Okay, so now let's talk about light-independent reactions, also known as the Calvin cycle. So first of all, where do light-independent reactions happen? So these steps take place in the stroma, and this is the inside fluid part of the chloroplast. So this is outside the thylakoids. So light-dependent reactions happen within the thylakoids, and the stroma is outside, but in this case, we will be outside of the thylakoids with the stroma. So the goal of the Calvin cycle is to use the energy made in the light-dependent reactions, so your ATP and your NADPH, and we're going to change carbon dioxide into glucose, and your glucose is your sugar. So these will be entering the Calvin cycle. So as you can see here, we're going to be changing this carbon dioxide, and we're going to be trying to making it into sugar. So the first step is going to be carbon fixation, as you can see here. So what is carbon fixation? So carbon dioxide from the air is going to be entering the cycle, and it will be attached to a 5-carbon molecule. And this 5-carbon molecule is going to be, so your CO2 is going to attach to a 5-carbon molecule, and this will form a 6-carbon molecule. And this 6-carbon molecule is unstable, and therefore will turn into 3-carbon molecules. So this is going to attach to a 3, 5C, and I'm separating how many there are. And since these two attach, it will become a 6-carbon. However, this is unstable, so we're just gonna make it into we're just gonna flip it so to make it stable. So this will become 6C. There you go. So yeah, we're just gonna try to make it stable by turning it into 2 3-carbon molecules. So we had 3, this becomes 6. You multiply by 2, because you had your 3 molecule. So now let's go into our second step, and this is going to be reduction. So in reduction, each 3-carbon molecule is going to be modified by ATP and NADPH, and this will form something called G3P. So you're going to be turning this into your 6,3C. So your 3-carbon molecule, and you're going to be modifying all of those into your G3Ps. So for every 3 carbon dioxides that enter the Calvin cycle, you will end up getting 6 G3P made. So that's why I wrote the 6 here. And then one of the 6, sorry, I just dropped my pen. One of the 6 G3P goes on to produce a glucose, and the other 5 are recycled to form your original 5-carbon intermediate, is what it's called. So your NADPH is going to go back into your NADP, which is positive. And remember that each G3P is glucose. So every time I refer to glucose, you can make sure and remember that this is your G3P. So as you can see, we have 6. So these would be 6 glucose molecules. So now let's talk about the last step, regeneration. So your last step is going to be regeneration. So 3 more ATP is going to be spent to convert these 5 G3P molecules that I just got that were made because of reduction in the last step, and we're going to convert this into 3 of the 5-carbon intermediate molecules from the beginning of the cycle. So as you can see, we're just going to be bringing it back into your 3,5C, and the 5-carbon intermediate molecules can then be reused again for the cycle to happen. That's why it's a cycle, because you can just keep doing it over and over and over and over again. So at the end of the day, this will bring your ATP back into ADP plus this, so that it can go back into your light dependent reaction, and then that will just bring more so that it can go all the way back to light independent. So at the end of the day, photosynthesis is basically a cycle. You'll keep going back and forth with the light dependent and the light independent reactions, and this at the end of the day will form your glucose. So that is how light independent reactions work. So the Calvin cycle needs to happen many times to make one full glucose molecule, and that one full glucose molecule will be used by the plant or in this case the plant, and it'll be used for energy and building material. So finally, let's talk about the importance of enzymes in photosynthesis, because enzymes are super important because they help all the chemical reactions happen quickly and correctly. For example, in the Calvin cycle, there's an enzyme known as rubisco. So rubisco is really important because it attaches carbon dioxide to your 5. It's going to attach it to your 5-carbon molecule, and in the first step. And without enzymes such as rubisco, photosynthesis would be too slow, and it wouldn't be able to keep life going because you wouldn't have enough energy. So at the end of the day, remember that light-dependent and light-independent reactions are especially important to make sure that photosynthesis occurs. You cannot have only one, you need both. And it helps plants, algae, some form of bacteria survive. And yeah, thank you.